Is it working? I'll just sit right here.
Yeah, okay.
All right, welcome everyone. Taking photos with movies.
That's after.
I think we should do it after, right?
Actually, if it goes really badly, the photos are going to
Oh, I see. Okay. Before and after. Okay. Hint, hint. Right? All right, welcome everyone. It gives me great pleasure to introduce Rob to you for his PhD thesis defense. He's going to be talking about his research in the next 30 minutes or so. So on the technical side, the way these things go, the first is going to be a public version of it, and then we're going to spend more time just the committee with Rob at the end. And then we're going to take the famous photo afterwards, just to contrast with this photo that we just took today. So he's going to be talking about his research, and because he's going to introduce that by himself, I'm going to give you some background on Rob on non-research statements. So Rob has been he joined in one of the first years in the Bioinformatics Ph.D. program, and for those of you that are in the program or are soon to join in the program, most of the things that you see in the program, in the structure of it, Rob has had some say into them, both in terms of the student retreat and all sorts of meetings that you know are currently part of the bioinformatics program, and that's also true for our lab. Rob joined our lab, a couple of months before I moved to UCLA. So Rob, Rob technically was set to join John's lab, I guess in 2012, and then fortunate for both the lab as well as Rob, we joined in 2012, and that's when Rob started working in the lab. And in the past year, because he learned everything that he could learn from my group, he joined, he's co-advised by Eleazar, and he worked in Eleazar's lab in the past year or so. So without further ado, I guess it's
my turn. Start with the password. There we go. All the secrets. First of all, I'd like to thank all of you guys for being here today. It means a lot for me to have you guys all here. And basically, I've worked with all of you throughout my time at UCLA, so I'm really glad you could all make it as I present a summary of my dissertation work here. I'm going to begin actually with a brief background of genetics. Our human genome, we can think about this as a 3 billion base pair long string of nucleotides that we can represent with A's, C's, T's, and G's. As humans, we're diploid. This means that we carry 2 copies of the human genome within us, one from each of our parents, and we can refer to these as haplotypes. If you look along our haplotypes, however, you'll notice that not every position in them is the same. There are a lot of locations which we call single nucleotide polymorphisms. These are locations where the genome can take 1 or 2 different alleles. For this first instance, there's a G allele and a C allele. One question we can ask, however, is how similar is my genome from the genome of other people? And it turns out it's 99.9% the same. We're really similar. However, that 0.1% of differences, those are what drive the traits and the disease susceptibilities that we can inherit from our parents. And so it's really important to understand what's going on with those. In the work that I'm going to present to you today, I'm going to focus on 3 different parts of understanding these differences. Their frequency within populations, how they're geographically distributed, and what biological function that they have. So my first set of projects that I'm going to present have to do with the frequency and geographic distribution of genetic variation. So we're going to start with this one that looks at ancestry. Thanks to a lot of increases in technology that allow us to more easily sequence individuals and read their genomes, we're able to survey many populations to actually figure out what the genetic variation looks like in each of these populations. When we do this, we can calculate what I call continental specific variants. These are variants or SNPs in the genome where one of the alleles is only present in one continental population. For instance, perhaps a C allele that's observed in Europeans but never observed in Africans or Asians. When we look at the 1,000 genomes data that we have publicly available, we find that Europeans, on average, carry about 14 of these continental-specific variants in each megabase of their genome. Africans carry about 110, and Asians about 12 in every megabase of their genomes. These are important because they can tell us a lot about ancestry. So some of us, like myself, my ancestry is relatively easy to understand. It's European. However, other people have much more complex ancestry. And as geneticists, we want to know not just what the proportions are, but what does it actually look like down at the genetic level. So to understand what's going on, we look at what's called the admixture process. When two populations that have been separated, historically either for political reasons or geographic regions, come together and mix, the genomes of their progeny, we say that they have admixed genomes. What that means, and every 2 bars here represents one individual, but what that means is that due to the recombination process, their genomes are actually a mosaic, where different pieces have different continental ancestry. And what we really want to do is we want to understand, if I look at one specific location in the genome, did both copies of the genome at that position come from population 1? Did they both come from population 2? Or did one of the haplotypes come from one population and one from the other? So to do this, we're trying to infer what we call local ancestry. Now, the problem is this nicely painted true ancestry at the top is not how we read the genome. It's not color-coded for us. All we get to know is that sequence of A's, T's, C's, and G's. So what we need to do is we need to find out, is this portion of an individual's genome, is it from the green population, or is it from the blue population? So one of the established approaches for doing this is called LAMP-LD. And it's representative of a lot of the approaches that exist. They use genotyping chip data, which gives us access to common variation. And I'm going to present LAMP-LD because it's one of the best performing methods. And how it works is quite intuitive. Essentially, you look at the sequence of A's, T's, C's, and G's in one individual, and then you compare it to haplotypes that you've gathered that are representative of the ancestral populations. For instance, you could get a lot of, for like population 1 and 2, those could be Europeans and Africans, in which case you can go to Europe, find a number of individuals who, like me, probably have about 100% European ancestry, and use my haplotypes to represent Europeans. You could do the same thing for an African individual. Then what you do is you simply take one portion of an individual's genome and you compare it to that same location in the haplotypes that you have, and you basically ask, which one's it most similar to? So as computational people, we clearly aren't going to eyeball this. We're going to do this with a hidden Markov model. So this is the LAMP-LD model, and it breaks the genome into a bunch of smaller hidden Markov models. In these, the nodes represent the different SNPs that we observe, and they're going to emit the different alleles. When we look across the entire model, it represents an entire chromosome of an individual. And the method LAMP-LD works chromosome to chromosome. So what we do then is we can take the entire genome of an individual, or the entire chromosome from an individual, and we run it through that model. And we're asking, which of these little hidden Markov models has the highest probability of emitting the sequence that we observe in an individual? And then we can follow that path through the Markov model, and from that infer the ancestry. So when we do this, we basically have a very good guess at what the local ancestry is, and that's our inferred local ancestry for an individual. But as you can see right here, it's not exactly what the true local ancestry is. While this method's very accurate, one of the questions we need to ask is, can we do better? Can we make a more accurate method, and also a method which is faster? Because this one can take a long time to run. So that's what my first project's looking at. It's asking, rather than using genotype chip data to do local ancestry inference, can we use genome sequencing data to make a better method? And since I'm presenting this to you, obviously we can. So we're going to start at the same place. We've got this genome sequence, and we want to infer the local ancestry. However, what we have as well, a rare variation. And a lot of these rare variants are the continental specific variants. So in this case, This C allele, it's going to be observed in population 1, but never in population 2. So if we see it, it tells us a lot about the ancestry of the individual at that specific location. So we're going to make a plot where we can plot the locations of all the CSVs and what population they come from. When we do this for the full genome, it looks like this. So if we look at it, what we can see is that sometimes we have these long strings of just these blue CSVs from population 1. And the reason we're just seeing those is because it comes from an ancestry segment that has 2 copies from population 1. And so when we see that sort of string, we can infer that both copies of that both haplotypes must come from population 1. When we're seeing a mixture of CSVs from both populations, we can infer that the true ancestry has a haplotype from population 1 and a haplotype from population 2. We can do the same thing when we just have population 2 CSVs, and continue for the entire genome. Of course, we don't want to be eyeballing this as well, so we're going to formalize this in a hidden Markov model. This model is going to have 3 states. Each of the states represents the different possible ancestry combinations. So the blue represents 2 copies from population 1, the green 2 copies from population 2, and the orange 1 copy from population 1 and 1 copy from population 2. What we're going to emit from each of these states are simply the observed CSVs. So when we compare that model to the LAMP-LD model, as you can see, it's much simpler and much more straightforward. And because of that, it can run a lot faster. We're going to leverage this increase in speed in order to do something novel with our method. We're going to make it sample aware. What this implies is that once we infer the ancestry for one individual, we can actually use those inferred results in order to make our method more accurate for inferring ancestry in other individuals. How we do that is by correcting errors in the CSVs. So if you look at these different CSVs that we observe, those are probably just mistakes due to like a small amount of training data. So like there's no way we should be observing a CSV from population 2 in an ancestry region that only has ancestry from population 1. So what we can do is we can remove all of those, and not just for this individual, but we just won't use those CSVs when analyzing other individuals as well. And this, we hope, will increase the accuracy of our method. And it turns out that it does. What I'm presenting to you here is a plot where the inference accuracy is on the y-axis. Each of these 3 sets of bars represents increased numbers of simulated individuals, starting with just 50 on the left and 1,000 simulated individuals on the right. And then we're going to simulate individuals from 3 different populations. The blue is African Americans, the green Puerto Ricans, and the red Mexicans. And as you can see, as the number of individuals we simulate increases, so does the accuracy of our methods. And that's because we have more and more individuals that we can use to correct mistakes in our training data. What's really exciting is that once we start getting to the larger population sizes, this method actually has better accuracy than the established methods, and it can run in substantially less time.
Well,
yes?
Is the reason that the African Americans start strong and stay strong is because there are so many rare variants to start with, or ancestry-specific variants?
Essentially, yeah. And Africans have about 110 per megabase. So
tenfold, no.
Exactly. I'm not going to present it here, but we actually do look at subcontinental admixture, and the accuracy is much less on that, but we're actually still able to do it a little bit, like separating like Germans from Finnish. And yes. So the reason this local ancestry is important and to make sure we have the most accurate methods is because we just don't want to understand the local ancestry of an individual, but we actually want to use that information. 2 other projects that I was involved with that I'm not going to present to you are using local ancestry in order to find lipid variants in Latinos, and also for leveraging local ancestry in order to look for gene-gene interactions. The next project that I am going to present, though, also uses frequency and geographic distribution, but it's going to apply it to understanding monogenic disorders, specifically for trying to identify what genetic variation is causing a monogenic disorder. So at its simplest, a genetic variant somewhere in the DNA in one of the protein coding regions can cause the complete disruption of a protein. Monogenic disorders occur when that disruption of the protein is enough to cause the disease. So some examples of this are sickle cell anemia, Tay-Sachs, and cystic fibrosis. These are things that a lot of people have heard about because they're relatively common in populations. However, the vast majority of monogenic disorders only occur in a small handful of individuals each year. And there's 2 examples of that. A lot of times, however, you might actually only have one individual with one of these disorders, and that's a huge problem from the statistical standpoint because sample sizes of 1 aren't very good for inferring anything. And so we can't use a lot of the methods that we might use for a more common disease, such as like diabetes. The biggest issue here is that one variant might be completely responsible for this disease, but when we look at the protein coding region of the genome, which is called the exon or the exome, individuals carry between 20 and 24,000 variants in that region. So somehow we have to get from this 24,000 variants down to like a single causal variant. So while this is a very long drawn out process, some of the first steps is simply to try to get rid of variants that we don't think could be the causal. So these diseases are very rare. So if the causal variant were common, then lots of people would have it. So that doesn't make sense. So what we can do is we can start by filtering out any genetic variant we see that has a frequency above 1% in a population. Then after that, we can use other methods such as polyphen, which give deleteriousness scores, and any variants which are predicted to be non-deleterious, we can also remove from consideration. Even when we do this, however, it still leaves hundreds, if not thousands, of variants that need to be followed up on. And that can take a lot of time, effort, and money. So we're going to ask if we can do this process better, and we're going to focus on this 1% frequency filtering step. So one of the questions is 1% where? So if I have, say, a European individual who has a monogenic disorder, should I filter out any variant which is observed 1% in Europeans, or should I filter out variants that are observed above 1% frequency in any population? Well, if we assume that the disorder is rare everywhere, then we should also assume that it's going to have a rare frequency everywhere. So as you can see, if we just used Europeans, we would include this particular SNP. But if we look at Africans, then clearly the SNP is above that 1% frequency. So what we really want to ask is this variant observed above 1% frequency anywhere? And if it is, we'll disregard it. We run into this big problem, however. And the problem is a lot of these frequencies that we're estimating, we're doing that with very small samples from certain populations. And because of that, there can be a lot of noise in our frequency estimates. And so even the true frequency of a variant in a population is well below 1%, we might observe it at like 2 or 3% just due to random chance. And so we really want to make sure that when we're looking at lots of populations, they're not accidentally filtering out our true causal variant. To do that, we develop a way to calculate a threshold. Our threshold is going to be T. And what this formula tells us is that if we look at a population and in that population we filter out all variants above some threshold T, it tells us the probability of filtering out a variant that has a true frequency in that population below 1%. So by using this formula, we can make sure to control our false negative rate, or our probability of disregarding the true causal variant due to random noise. So we're going to take this data, and we're actually going to look at some real individuals who have monogenic disorders. Our collaborators provided us with the genomes of 20 individuals with Middle Eastern ancestry. Importantly, we actually knew the causal variants in these individuals for their disorders, and so we can make sure that we weren't going to be filtering those out. And then there's 3 different filtering strategies that we're going to use. The first is called no ancestry. This basically takes all of the individuals in the 1,000 genomes data as well as in the exome variant server, and it calculates the allele frequency just across all of those without taking populations into account. When it does that, for instance, if we look at the recessive disorders, that leaves on average about 60 variants per individual that need to be followed up on, or about 750 when you're looking at a dominant disorder. Another strategy is called pop matched. This basically says if we have a European individual, let's calculate the frequencies based on Europeans. When we do that, we actually see a reduction down to about 40 variants remaining per individual for the recessives and 600 for the dominant. However, when we look at all the populations that we have available and use this thresholding approach, what we see is that we can get all the way down to only 30 variants remaining for the recessives and about 450 for the dominant. So that's almost a 50% reduction in the amount of variants that need to be followed up on. And this can save a huge amount in terms of time and labor costs for researchers to identify which of these variants is actually the causal. Importantly, we can do this, and we can do this making sure we're not filtering out the true causal variants. And we know with these 20 individuals that our method never did filter out any of the true causal variants. So at this point in my dissertation work, I actually make a shift from looking at the frequency and distribution, the frequency and geographic distribution of variants, and trying to understand how we can use those two to understand human traits, and start looking at the biological function. Specifically, I'm going to be looking at gene regulation, and I have 2 projects I'm going to go into in depth with you, and then I'm just going to briefly touch on the haplotype-based EQTL mapping project. So in my first one, so these are all dealing with gene regulation. What that basically means is we're looking for genetic variation that can either increase or decrease the rate of transcribing DNA into RNA. So one way we can think about this is with a simple additive regulatory model. In this, we've got some regulatory element. It could be a transcription factor binding site, for instance. And then we're going to have one SNP that has an effect on that. And we're going to represent, and that SNP, the genotype of it is either going to be 0, 1, or 2, representing the number of alternate alleles that an individual carries. So as you can see, in these 3 individuals, if you don't carry any of the alternate alleles, you're making essentially 100% of the mRNA. If you have one of the alternate alleles, you'll make 50%. And if you have both haplotypes affected by an alternate allele, you don't make any. This is, of course, a very extreme example. Very, very few, if any, SNPs that regulate gene expression do this. Most of the time, it's a much smaller effect that a SNP will have. When we do have a SNP that has this sort of regulatory effect, we term that an expression quantitative trait locus, or an EQTL. And then any gene that has one of those EQTLs, we call an E gene. So one question we can ask, though, is this model was with one SNP. What happens, however, if we have 2 SNPs that can affect this regulatory region? Well, it becomes a much more complicated model that I'm going to work through with you. So first of all, you can see that in an individual that doesn't carry any of the alternate alleles, each haplotype can still produce all of the mRNA that it normally would. So it's fully expressed. However, in individuals that have one of their haplotypes affected by either of the alternate alleles, or in the far side, by both of them, they're only going to make 50% of the amount of mRNA they normally would. In these 3 individuals, both of their haplotypes are affected, but each time at different SNPs. In this case, these are going to have no expression, so they'll make 0% of the mRNA. So one really interesting case of these, if you look at the far right, are these guys, because the genotypes are the same. Both of them have a genotype of 1 because each SNP carries 1 alternate allele. However, the phase of the alternate allele is actually going to impact the amount of expression of that gene in these individuals. So what you can see is none of the SNPs are actually the best predictors for the expression of this gene. What is, however, the correct predictor is simply the number of haplotypes that are affected by alleles. So we're going to call this a compound regulatory predictor, or a CRP. And as you can see, the CRP perfectly correlates with the amount of expression. So we're going to delve into this a little bit and look at this through a graphical model. So what we have is we have this CRP affecting the expression level. This CRP, however, is composed of 2 SNPs. And these SNPs are going to be correlated with that CRP. They're also going to be correlated with each other potentially through linkage disequilibrium. So when you do a normal EQTL study, when you're looking for them, what you're doing is you're trying to find an association between the SNPs and the phenotype, which is the expression level. So when we do this, we're not actually testing the true predictor, which is the CRP. And what that means is that this test, testing the SNPs is actually going to have reduced power relative to the test of the true predictor, which would be the CRP. How much that power is reduced by is going to be a function of the correlation between the SNPs and the CRP. If that correlation is very high, it means the SNPs are tagging the CRP, and your test might still have the most power that it could. But if that correlation is very low, it means the SNPs are going to be very bad tags, and so you're not going to be able to have a significant association when testing the SNPs. So we need to understand this correlation. So what I'm showing you in this plot is the allele frequencies of the two SNPs. So the x-axis is the allele frequency of the first SNP, the y-axis is the allele frequency of the second SNP. And then the color, that represents the maximum of the correlation of these two SNPs with the CRP. So when you have like the dark red color up there, that means that at least one of the SNPs is very good at tagging the CRP. Whereas when you look at like the yellow, the green, and the blue, those are locations where neither of the two SNPs has a strong correlation with the CRP. And it's in that region where we're not going to have the ability to call e-genes with this regulatory architecture, because we're going to have low power with a SNP test to have an association. So what we propose doing when we do an EQTL study is both testing the SNPs for association and testing the CRPs for association. The problem, however, with testing every possible CRP is that's a huge multiple testing problem. So what we have to do is we have to do a selection procedure. First of all, we're going to recognize that if we test a CRP that falls in like this red section up there, it's really not going to give us more information because one of the SNPs is already so well correlated with it. So we're not going to test those CRPs. We're only going to test CRPs that follow in the yellow, green, or blue regions up there. The second thing is a SNP will have reduced association due to the correlation. It won't have eliminated association because it can still be induced due to the correlation. So we're also only going to test for CRPs with SNPs where both SNPs show some like marginal significance level. So we're going to evaluate the power of this combined SNP and CRP test, starting by using some simulations. The first simulation we look at is a null simulation. For this and for all of the other different architectures on here, what we do is we simulate 18,000 genes, which is going to be similar to the real data set that we're going to apply our method to. And we run our test, we calculate an empirical p-value using permutations, and then we control the false discovery rate at a 0.05%, and we report how many e-genes we find. However, before I get into that, I do want to note that with the null simulation, while it's not up here, it does control the type 1 error rate at a 5% level. So our methods are well calibrated. So as you can see in the null model, we don't have any significant genes, which is what we expect to see. And the next one to look at is when we implant a single causal SNP, and this is a common SNP. We simulate this such that the SNPs explain 8% of the variance in the gene expression levels. When we do this, you can see that our method, compared to both the SNP test and a SNP and interaction test that we compare to, has essentially the exact same power. The number in parentheses over the 7, that shows the number of genes which weren't in the same set of E genes. So there were 7 genes found by our SNP and CRP test that aren't in the same set of genes found by the SNP test. When we look at a lot of the other causal architectures that we simulate, the CRP architectures, as well as when we simulate 2 causal independent SNPs and 2 SNPs that have an interaction effect, you can see that our method finds significantly more genes than the SNP test and the SNP and interaction test. So what this is really telling us is that our method isn't necessarily just picking up on CRPs, but it's actually going to be picking up on any causal architectures where there's an up or down regulating effect due to any possible more complex architecture than just a single causal SNP. So we're going to take this method and we're going to apply it to the Giavotti's gene expression data. We're going to use the European data that they provide, which includes 373 individuals and 18,621 genes. They also provide the full sequencing data for these individuals. So what we do is we we computationally phase the genotypes so that we can compute the CRPs for each individual. By running our test on those, what we find is that the standard SNP test finds 3,500 E genes, whereas our test finds 3,529. This is only a small increase in power, but that's actually expected. And that's expected because one of our CRP selection criteria is that we're only going to be looking at SNP pairs where the pairs already have near marginal significance. And so we're not expecting a large increase. However, we do find some interesting things when we look at them more closely. So of those 3,529 significant E genes that we have, we ask, do they have a significant SNP association, a significant CRP association, or do they have both? And it turns out that 153 of them only have a CRP association. In those genes, we can ask how much of the variance is explained by that CRP association. It turns out that about 7.1% of the variance in the trait on average. Whereas when we take the top SNP from those same genes, it's only explaining about 5%. So the CRP is, since only the CRP is significant, we are expecting this, that it explains more of the trait. However, when we look at the 2,222 E genes where there's both a significant SNP association and a significant CRP association, in 37% of these, the CRP association has a stronger signal. And when we look at that, that CRP is explaining 12.6% of the variation, whereas the top SNP is only explaining 10.8. What this means is that whatever the true underlying regulatory architecture is, the CRP test is better able to capture that than our SNP test is. I'm not going to show you anything about when there's no CRP because basically all the numbers are identical because the CRP doesn't have any, it's not inflating the statistics at all. So that was the first method here. The haplotype EQTL-based haplotype-based EQTL mapping increases power to identify E genes. What I do in this paper is I actually extend the CRP idea. Because what we learned there is that a lot of times that there are different haplotypes that the CRP is tagging, which we're getting better from when the architecture is not just like a single causal variant. And so rather than looking for CRPs explicitly, we're just going to look in that manuscript for haplotypes where not all the haplotypes in a region have the same effect size. And that manuscript's currently submitted at Recom Genetics, so hopefully in a month I can present that all to you if that gets accepted. What I am going to focus on now is another manuscript which is also submitted at Recom Genetics, and it's a method called ggmend. So when we were looking for CRP associations, we were looking for locations where some genetic variant had an effect on regulation. In ggmend, what we're going to look for is when the expression level from one gene is actually able to impact the expression level from another gene. So an easy model to work through this is we're going to have a genotype of one gene represented by G1 and expression levels for that gene represented by Y1. However, pretty much all genes are also regulated by unobserved confounding factors. These can be environmental things, other genes. We don't know because we can't see them, but they are going to have an effect on expression levels. However, there might be another gene that has its own genetic variation affecting it and its own expression levels, and that can also be influenced by that same unobserved confounder. When this occurs, there can be a correlation between Y1 and I2, not due to a causal effect of one gene on the other, but simply due to a shared confounder. So this is going to be our H0, our null model. What we want to do is we want to distinguish this model from our H1. In this model, it has the same effects of genetics and the confounding effects, but also the expression level from the first gene is able to influence the expression from the second gene. So the big problem in finding this is due to that confounder, we can't just regress Y2 on Y1. Because if we do that, we might observe an effect, but we can't tell if that effect is going to be due to the expression of Y1 or if it's just due to the unobserved confounding effects. So what we're going to do is we're going to draw on some insights from Mendelian randomization. What that means is that we inherit SNPs randomly from our parents, and which SNPs we inherit isn't going to be due to any confounding effects, so that we know that the genotypes of these in these models are independent from our confounding factors. When we do that, we can use G1 or G2 as instrumental variables. And we can estimate these different effect sizes. Or we can estimate the effect of G1 on Y1, which is our beta 1, and that's independent of U. We can regress Y2 onto G1 and to see if this SNP has any effect on the expression level from the second gene. What's important is this regression here, its estimated value is going to be independent of U because there's no backdoor paths that connect G1 to U. When we take the ratio of those numbers, the beta G2S1 divided by beta 1, that's actually going to equal an estimate for the effect of Y1 on Y2. Once we have that estimate, we can use a permutation analysis which breaks this value here. And by doing that, we can establish a p-value for how significant that gene-on-gene effect is. In order to see if our method can actually find anything in real data, we're going to use the genotype tissue expression database, or the GTEx data. There's a lot of tissues available in this, but we're going to take the 4 that have the largest sample size, which are blood, tibial artery, skeletal muscle, and adipose. And we're going to see if in any of the gene pairs in these tissues, if we can observe a significant gene-on-gene association. Again, we have a really big multiple testing problem because we're looking pairwise. So we're going to restrict our analysis to only looking at genes that have a very, very strong cis effect. And then also where the genes already show correlation. But we don't know if that correlation is from a confounder effect or if it's due to one of these gene-on-gene effects. But we're restricted to that because if they don't show any correlation, then we know they're not going to have any gene-on-gene effects. So when we do this, these are the results that I get. And what I'm presenting to you here is the top result from each of the different tissues we look at. Once running all the tests, we do FDR control. So there's a 5% false discovery rate in each tissue. So if we look at tibial artery, we do find one gene pair in there where there's a significant gene-on-gene effect. It has a p-value of 0.05 before we do any correction. And this gene is significant when we control the FDR at a 0.05 rate, and it would still be significant if we were controlling FDR at a 0.02 level. When we look at skeletal muscle, we find one p-value, which is marginally significant, but it just barely doesn't pass the FDR control of 0.05. For blood and adipose, we don't observe any significant, or we don't observe any gene pairs there that pass the FDR control. And that brings me to the end of the work that I've done for my dissertation. Here's a list of the papers that I'm first author or a co-author on, and the bottom 2 are currently submitted at Recom Genetics. And I hope to present both of those to you in more detail here. And I have a lot of people to thank for helping me on all of these projects along the way. First and foremost, Bogdan's group. They have been with me basically since I've joined the lab. And they are an outstanding group. And I'm always impressed by just how much insight they have into the genetics and into the statistics. And there are so many of them that I turn to all of the time when I need help with something. And they're always willing to take the time to help me through my projects. I've presented this to them multiple hours in the past little while, and they always make time for me. And that's extremely important. And also, they're almost always there all the time in lab, and we have a great lab culture. And I think one reason I've really enjoyed my time at UCLA is because I'm always surrounded by friends. And I'm very happy to not be moving very far away from them all here in a little while. I'd specifically like to thank Wen Yung. He and I were basically lab mates by ourselves in a very large open lab for the first little while as I joined the lab. And then everybody joined in after that. And while I'm only co-authors with a select few of them on my manuscripts, all of them have contributed multiple hours of their time at the whiteboard, helping me work through math or helping me polish my presentations to make sure that what I say actually comes out clearly and intelligibly. Zara's lab, who I joined a year ago, I've been very happy getting to know them more and to seeing their work and working with them. We just got back from the Recom conference in Hong Kong, which was really fun, and they are a great group of people and also very good at the work that they do. And in the past few months, they've really made me feel at home in that lab and included me, and I'm very grateful for their efforts there. I do have a shout out for Kirk's lab. For the first 2 years or so in Bogdan's lab, we had joint lab meetings with Kirk's lab, and he had a large role in, the people in his lab also had a large role in inspiring my first couple of projects. And I still think a lot about population genetics because of their influence on me and how to use those insights from population genetics to make more powerful methods. I would like to thank Hain Lee, Eleazar Eskin, Bruno Reversade, Stan Nelson, and John November, who are my collaborators on some of my papers, who I'm not going to mention in other acknowledgments, but I do want to say that I'm very grateful for them and that they took the time to sit down and help me understand the monogenic disorders, how to look at them, how to read the data, and what all meant. My monogenic disease paper definitely would not have gotten in if it weren't for their help in assisting me doing that. I have a lot of friends and family who might not always contribute to my scientific advancements, but definitely contribute to my emotionally being able to work on my scientific advancements. So Pete, Larry, and Chelsea, Pete and Larry came in my first year of bioinformatics class when we were still a very wee program, and have remained good friends ever since. And it is always outstanding to see them. Chelsea was in Czar's class with us and sat next to us, and then we did a lot of homework together, and we had these great projects where when for your final projects we would all go to Chelsea's place and we would order pizza and sit there and just code for like hours.
Is that considered cheating?
No, because we all worked on different projects. But so they're really great. The next group of people, in addition to being good friends, Ashley, Arthur, Rebecca, Kim, Adriana, and Kiku, they have taken on a lot of the projects that I either co-started with them or started before their time and have been continuing them, such as the student retreat and the student meetings and stuff like that. And they've taken on leadership roles within the program, and I'm very grateful for their efforts, because one of the reasons we have a great program and we have great seminars and we have great opportunities is because of the leadership that they've taken on, and that's definitely helped me become a better graduate student. Elise, Sean, Catherine, Taylor, Christian, and Rob are all good friends of mine who sometimes when I don't feel like doing work, we go to the pool and get some laps in at noon, and those are the people who I could always drag with me to do that. And they have been great. Elise also does some backpacking trips with me on the weekend, and they're a lot of fun, and they're the people who I get to take my breaks away from work, because that's a very important part of graduate school. And I am grateful for their always being friends with me. Then I have my family, who for some reason believe that my wife is the one who earned this degree and have reinforced this on me many times. They're coming here in a week and apparently brought gifts for Rebecca. I don't know why, but I do also appreciate the fact that before I got married, they supported me in my efforts. And I've had many adventures before deciding to go to graduate school, and they've always supported me through all of the twists and turns in my life. And I probably wouldn't have actually enrolled in graduate school and gone down this process unless I knew that I had a great family to support me. And then finally, my wife, Rebecca, who, as I said, apparently earned my degree according to like all these people I'm talking to. But no, she has been great. She has always been there for me on days when I've had good days to listen to me, on days when I've had great days to sit and stare at a piece of paper as I put math equations down on it, as she nodded along and tried to understand. And to her credit, she's actually very good at understanding them. So I do appreciate all that she has done to support me along the way. Then I have my committee members, Rita, Janet, Kirk, and Eleazar. I actually knew Rita and Janet long before my committee from classes that I had taken. And I have always been impressed with not only their ability to teach, but how much time and effort they put into the students in their class, making sure that they understood the concepts, making sure that that their students felt supported and that they could achieve in the things that they did. And they also, along the way, gave a lot of advice on how to navigate graduate school and the things that you should be paying attention for. And I'm very, very grateful for all of that. Kirk, as I've mentioned before, we had joint lab meetings with him, and he has done a lot, both in terms of helping me understand genetics, but also in terms of getting advice from him as well in the grad school project and in how I should make the decisions that I do. Eleazar is for the first few years that I knew him, he was very much my yes man. It's like, hey, we want to do a retreat. He's like, yeah, do it. And it's like, so a lot of the ideas that we came up, we would go to Zar and be like, can we get permission to do this? Like, who should we get permission from? Let's go ask Zar. Zar will give us permission. And so I'm very grateful for that. He helped me spearhead a lot of my extra scientific efforts. And that's been great. It's helped me feel like I've very much been a part of the program and that I could communicate with faculty. And I think that's really important, because if we don't have that sort of communication happening and as graduate students being able to help shape the program that we have, then we're not able to smooth out the rough edges. And I'm grateful that Zar was always willing to listen to ideas that we had and encouraged us to go down that. Also, as a co-advisor now, he has great ideas and is very good. He has definitely taught me how to get papers out really quickly. If nothing else you should learn from Zar, you should read his blog about how to get papers out really quickly, because he has good strategies for doing it. The cheat sheet is you write your paper before you do any work, and then you just have like tables you have to fill the blanks in on, and then you submit. So, but no, he's been a great advisor for this last year. And then finally, my advisor for the vast majority of my PhD is Bogdan Pasaniuc, who I still don't know if I'm saying his name correctly or not. And the reason isn't because I don't sometimes know how to say his name correctly, but because I hear it mispronounced so often that I forget what the correct pronunciation is. And everybody asks me how to pronounce it whenever he's about to give a presentation or anything. So this was actually the hardest to think about what I wanted to say about Bogdan, and I realized the problem was how I was trying to phrase it, because I was always like, Bogdan's outstanding at this, or he's the best at this, or what really defines Bogdan is that, and I was trying to find one thing he's really good at. And so I'd always find one thing he's really good at, and the problem, of course, is well, no, because that doesn't talk about this other thing he's really good at. So it turns out he's actually really good at a lot of different things, and so we're going to go through some of them. I would say the advice I give to students that are considering joining Bogdan's lab, I will now let Bogdan know. And it's that when you give your first presentation in Bogdan's lab, expect to never get through your second slide. It's just not going to happen. Everyone in Bogdan's lab is smiling because they all know it's true. So I think, in addition to the science, which we'll talk about here in a second, one of his greatest qualities is that he really cares about our ability to communicate our ideas clearly, to the extent that sometimes if you don't have a good intro slide to your months and months of work that you've just done, you just don't get through that intro. Because if people can't understand what you're trying to do, you're never going to be able to be a good scientist, because you have to be able to communicate your science to people. And not just in terms of our presentations, but also in terms of our writing style. We have drafts of manuscripts where we're on version 50 something. And it's because he'll take the time to go through and to read your papers and make sure they sound good, and to teach you how to phrase things better, and how to make sure that people can understand the ideas that you're trying to communicate. And I very much appreciate the time and effort that he put into doing that for me. Secondly, scientifically, I have had many a crazy idea in lab, some of which have conveniently become published. But Bogdan has always encouraged us in the lab, and he's encouraged me to take the ideas that I have and to run small simulations to see if they work and to really flush them out. And he's encouraged us to go and find the things that interest us in our academic careers and to put our time into researching them. And I'm very grateful for that, because I've always been able to find projects that I really like, because he gives us both the freedom to go and find those projects, but also he provides that nice, like, hard piece of metal to knock off the sharp edges of our ideas that are just really bad edges of our ideas, so that when it comes out, we actually have a nice project. And so his ability to take the ideas that we have and help us shape them into something that will be of value to the scientific community has greatly contributed to my ability to come up with ideas and then to figure out how to research them and publish them and get them out. After that, I also really like how Bogdan's always in the lab. He's making jokes. They're hilarious. All of them, every single one of them. But no, he's great to have around. His presence in the lab and his joking, I think everybody really enjoys being in his lab, and I think that's largely just because of how he always is interacting with us, and he's making us know that he cares about our daily lives and the things that we're involved with. And I really appreciate that as an advisor, and I've had a great time here largely because of your efforts and the ways you've run your lab and helped me learn about the science and how to write about it and communicate it to people. And yeah, and with that, I would also like to thank everybody else here who I may or may not have mentioned for being here and supporting me. It's been an outstanding 6 years that I've had here, and that is largely due to all of your efforts. Thank you very much.
Any questions on the admixture, the ancestry, like ancestry using reverse SNPs? What do you think the limit of it is in terms of your ability, what resolution can you get?
Yeah, so we actually did apply it to subcontinental admixture. So we looked at the 1,000 genomes populations, and we asked, can we distinguish one population from another population? And there's kind of two big problems with resolution on that. The first is it's highly likely that European individuals are already admixed, because using these country codes is kind of a like ad hoc way of being like, oh, well, you say you're from like, England. Like, are you really from England, or did you have like a grandparent, like a great grandparent from France or something? And because of that, when you train on like the English individuals and the Finnish individuals and the Italians, you might already have admixture that you're training on. And so that's the first problem. That said, for a lot of European countries, if you take like a 1 megabase region of their genome, you can, in like a phased haplotype of it, using the CSVs, you can call the correct country usually about 50% of the time in a 1 megabase region. So the other big issue that we have, and so this is correctable. So we have bad training data, but if we had enough individuals of European ancestry, what we could do is we could use this sample aware idea, and we say, okay, so people who are being called as from like Germany in this region, if we see like an Italian CSV, we can remove that CSV. So as we get larger and larger samples of individuals, we might actually be able to increase the resolution, essentially by trying to reconstruct better ancestry panels that don't have this problem of having admixture between countries or become regions. One other way to do it would be to forget about trying to assign country and basically use a PCA plot of Europe and divide the different populations up based on the PCs and ask how well can we assign a region of one individual's haplotype to each of these different like populations we see on a PC. And that I think we should be able to do relatively accurately if we just had more training data.
Have you thought about trying to apply something like that to like the 12,000 Chinese samples that took
Yeah, I have not thought about that, mainly because I haven't thought about this project until about 2 weeks ago when well, actually more like 2 months ago when I started writing my dissertation, but the
there will be like sequence cohorts of like tens of thousands.
Yeah. And this method should be very interesting when applied to those because it's very, very fast, and we should be able to relearn the within like country admixture much more quickly with it. Yes.
So Rob, can you say a bit more about allele frequency and the haplotype methods in CRPs? Were those more related to common variants, rare variants?
Yeah, so the original methods use genotyping chip data that looks at common variants, and basically the problem with those is that they kind of took these chips and they looked at a bunch of like European individuals, maybe some individuals from other countries, and they basically said, well, all of these individuals seem to have these SNPs, let's survey these ones. And they're not very accurate when you're looking at genetic variation that only occurs in like 1 or 2% of the population. And because of that, when they ran their methods, they were looking at haplotypes that based basically just on these common SNPs. When we get the sequencing data, basically we're able to read the full genome like that. And like, for instance, this C, it might be very rare. And what that means is either that it has once had a higher frequency and drifted down, but more likely it originated in some population, and then due to like random drift, the frequency of that slowly increased. But if that happened in, say, a Native American population, that variant isn't really going to be able to travel to Europe or Asia or Africa because there's a huge geographic barrier to doing that. And so what will happen is some of these rare SNPs will drift up to moderate frequency within one population. And so they'll be observed there in a lot of individuals, but won't be observed in other populations. Once the populations do start admixing, then that variant will start flowing into other populations, but it will be very, very rare in those populations, so likely we just won't observe it, if that was what your question was.
Yeah, I was thinking about more the gene regulation stuff.
Oh, you want to do it? Could you rephrase your questions in terms of rare variants and gene regulation?
Yeah.
Or yes,
Yeah. I mean, that was my question. Were the effects you're detecting more rare or common variants?
Yeah, so that's actually a really good question. In our simulations, we did simulate some rare variants and were better able to detect them, probably due to just some combination of common variants was tagging that rare haplotype. In the real data, it would make sense that there is a lot of rare variation, and that segregates on different haplotypes. And so when you start tagging haplotypes instead of SNPs, the CRP might happen to just be tagging the haplotypes that contain some burden of rare variation. And that's actually something I really want to look into. What I didn't talk about here is one of the reasons this is really interesting is because it's showing that there are a lot of effects that are better tagged by the CRP than they are by a single SNP. And what that means is rather than running a lot of the fine mapping methods that happen right now, we can start looking for fine mapping methods that are based more on haplotypes. And like these 4 haplotypes, for some reason, are upregulating in this region where these ones are downregulating. And so what's different about these haplotypes as opposed to looking at the correlation structure of more common variants, which is how a lot of the current fine mapping methods work.
Great. If there are no other questions, let's invite everyone but the and there's a reception at 5:30, and also now in the other lab. Yes. And there's another one at 5:30 in Boyer Patty.